Good morning, guys. So, we're getting close to the end. We're going to wrap up a little bit with the next couple of lessons here. So, um, one last thing I'll tack on kind of right at the end are GMOs, genetically modified organisms, because it's somewhat related to what we've been talking about with genes and gene regulation, and also relates to some real world stuff that you've probably heard about. In the past, I've talked about this more in depth, but obviously we don't have as much time for it this year, so it's going to be tacked on kind of right at the end. We'll do like a short little activity with it tomorrow, but we really don't have a lot of time for it, and there aren't finals this semester, so it's not like you're going to be asked to know that much about this anyway. So I'm going to go through the notes today. We'll look at some articles tomorrow, and that's mostly going to do it for this, just for the sake of how much time we have left. So, so we'll get right into it. This shouldn't be too difficult, and this should be pretty short, so I'm just going to go through this today. And like I said, we'll go through some article examples tomorrow. So, you've probably heard the term GMO before. If you don't know what GMO stands for, it stands for, wrong thing, genetically modified organism. This means that it is something, it's an organism that has had its DNA changed through non-natural methods. So what I mean by that is any kind of change that is brought on by human interference, whether that is gene splicing, like actually going into the DNA and putting in what you want, or even things like artificial selection, which we'll get to in a little bit, where we artificially breed for certain traits in organisms. That also counts as GMO. Well, depending on the context, but anyway. Types of GMOs. Like I said, the first type is artificial selection. We've been doing this for hundreds of thousands of years. You might not necessarily think of it as artificial selection, but what makes it artificial is that humans are selecting for the trait, the desired trait. You wouldn't see these traits being selected for in nature, which you can clearly see with some obvious examples. So for example, when we're looking at dogs, you know, some dogs are bred for advantageous traits. You've got your hunting dogs that are good at smelling, hearing, running, whatever it may be. You've got your show dogs that are good at competing and running and all that stuff, which would make sense in a natural setting. But then you've got your dogs that are bred for, you know, just to look cute and be a pet and a cute little toy dog. So you got your toy dogs, your chihuahuas, your terriers, your pugs, those kinds of things. So again, bred to be cute, bred to be friendly and fun, adorable, but put them back in a nature setting, and I don't think they're going to do too well. Not natural because it's caused by human interference with organisms' mating patterns. We also see this in livestock. So obviously, if you are working with livestock and you're working with breeding animals for market, you're going to breed animals that either produce the most meat if you're doing a meat market, or if you're into dairy and you're doing dairy breeding, you're going to breed cows that produce the most milk or the best quality milk, so on and so forth. You're going to take your best organisms and you're going to breed them together to get the trait that you want exaggerated down the line. Same thing with plants, which I'm going to get to in a second. If you're breeding crops, you're going to breed the crops that produce the most whatever part of the plant is that you're eating, whether that be seeds or kernels or maybe it's the stalk. No one eats the stalk, not the stalk. Whatever, you get what I'm saying. Whatever part it is that you're eating, you want to exaggerate the growth of it. Maybe you want to exaggerate the speed at which it grows, how tall it grows, the flavor, whatever it may be. You're going to breed for that kind of thing. And there's a really good example in a little bit. Oh, well, here's one right now. This isn't the one I was thinking of. This is another good example. Corn is a very good example of this as well. If you didn't know, in nature, the plant that corn was bred from was not as advanced as it was today in terms of like how many corn kernels it had. It actually had very few kernels on it. And over time, we bred for corn stalks that had more and more kernels because obviously that's the part that we eat. So the more kernels that it has on the stalk, the better, or on the ear, the better it is for you. The more you're going to have food, or the more food you're going to have, so the better it'll be for you. So over time, farmers would select for the plants that would produce the most kernels, and over time they started to produce more and more and more until we get the corn that we see today with all of the kernels on it. And you can imagine that. Another good example, I'll come back to it in a second, I don't think it's on this PowerPoint, but either way, don't tell Mr. Noonan, but again, artificial selection doesn't necessarily select for the best traits. Corgis are very short and stubby with very short legs, and while they're adorable, I don't think they're going to fare very well on their own in the wild. They might, maybe I'm wrong. But either way. The next example is probably the more sci-fi one, the one that you're probably thinking of when I say genetic engineering, or when I say genetically modified. So this is where scientists go in and they can actually splice in certain strips of DNA into certain organisms based on a number of factors, which we'll talk about. So what this process does is scientists have managed to find ways to isolate fragments of DNA, which we've looked at in the past. And then depending on the size of the fragment and also how well it can integrate into other fragments of DNA, sometimes you can insert that fragment into other organisms and have it express the trait that it normally would into organisms that you would normally never see it in. A really good example of this is the green fluorescent protein. Man, that's hard to say. Obviously, for named so for its green coloration and its glow, this is very similar to what we just looked at with the bacterial glowing. So we had the same idea. You've got a gene that produces some luminescent protein. Slightly different from what we looked at yesterday, though. But what you can do is, since this is a very small gene and a fairly simple gene, you can splice this into various organisms in order to get them to produce that green glow. Normally this is found in jellyfish, but we've been able to splice it into bacteria, worms, insects, and even small mammals like mice and rodents, mice and rabbits. You can actually buy, I'm not sure where, but I know you can actually buy some organisms that have this gene in them. My high school bio teacher had a tank of fish that had this protein in it, and if you turn the lights off, they would glow. So the reason why they do this, aside from just like looking cool, because obviously there's a market for people that are like, oh, cool, glow in the dark fish. It does look cool, but obviously there's more practical applications for it. The biggest thing that it's used for is as a marker, and what I mean by that is if you are splicing in genes, for example, that are, let's say you're looking at crops, and you want crops that are going to be more cold resistant, or crops that produce a chemical that is a natural pesticide. A lot of things like that are not a physical expression that you can see. Like if I make a crop that's more cold resistant, or if I make a crop that has a gene in it that makes it produce more cellulose or something like that, I can't see that on the surface. I don't know if that gene was actually taken up. I don't know if I was successful with my gene splicing. So what I usually do is, or what scientists usually do, is that they will attach whatever gene they're integrating into the cell to a glowing protein, so that way they know if the organism comes out and is glowing in the dark, they know that that gene was taken up, and if that gene was taken up, they know that the desired gene was also taken up, if that makes sense. A couple restrictions with this, because you can imagine, I'm sure some people's imaginations were running wild with this kind of thing. It doesn't work with everything for a couple of reasons, which I'll go into. First and foremost, the DNA of the host and the donor have to be similar. We looked at a while ago when we did our natural selection and evolution, we compared percentages of DNA that were similar between organisms. Some organisms are very similar, some organisms are very distant. Obviously, if you take certain genes from, let's say, a plant and integrate it into a human, it's not necessarily going to work. That's why you don't have cells with, or human cells with chloroplasts that can do photosynthesis. Even though I think that'd be a cool thing. So on and so forth. This is also why you can't make flying pigs, for example. Bird wings will not work with the amount of information that is in the DNA that is necessary to produce a fully functioning bird wing. Will not work with a pig for another couple of reasons. Second thing, you have to have a vector for getting your DNA into the host, and what I mean by that is you have to have a way from getting, so obviously these are DNA samples from two different organisms. You need to have a way from getting the DNA from organism 1 to organism 2. Most of the time this is done through a virus, because viruses naturally do that. So you take a virus and instead of letting it infect with the natural viral DNA, you replace the DNA in the virus with the fragment that you want, and then what the virus will do is the virus will go to the host cells and it will infect it, and it will inject the new fragment of DNA into the host cell, and then the host cell will uptake the DNA, because that's how viral DNA works. Anyway, it's a long process. We didn't really go into viruses this year, but that's not the only way. You can just do it through, I want to say needles, but it's like obviously a lot more intricate than that. But anyway, basically you have to have some way of getting the DNA from point A to point B. And then lastly, and this goes along with the flying pig thing, the gene can't be too big. If the DNA takes up a lot of space, remember, you're adding information into the DNA that was not previously there. And we just did a whole thing on mutations about what could happen if you insert DNA into places where currently existing DNA already is. You can imagine how much stuff you'd mess up. So the gene has to be small enough that you could squeeze it into a coding sequence and not have it mess up crucial genes. You can imagine, for example, going back to the pig example, the amount of genes necessary to make a fully functioning wing is a lot of information, and if you were to plop that down in the middle of someone's DNA sequence, you could knock out genes that are important for organ development, limb development, and vital hormone and chemical production, those kinds of things. That's going to really mess somebody up. Imagine you had a recipe book that was 50 pages long, and all of a sudden someone copy and pasted and slapped in 20 pages worth of a random recipe just somewhere randomly in the book. You could be in the middle of cooking something, and someone just randomly slapped in a brand new recipe for something else while you're trying to cook, and now you have to go through all of that before you can get back to what you're actually doing, and it makes a big whole mess. So here's some real-world examples. We'll look at a couple examples tomorrow, which is why I was kind of going to chunk this up into two parts. It's mostly used in agriculture, and you see this a lot already in society. If you've heard anything about it, some people are on the fence about GMOs, non-GMOs, and there are risks involved, which we'll get into tomorrow. We're going to look at, like I said, depending on people's stance on them. But if you go to the grocery store, I don't know how involved you guys are with grocery buying at your age. If you were like me, you just let your parents take care of it and didn't really worry about it. But if you have noticed, or the next time you go to the grocery store, look at the packages and you'll see a lot of them are labeled with GMO or non-GMO. Depending on legally, if it's genetically modified, they have to put it on the package. But a lot of places will also advertise non-GMO because a lot of people are looking for that and they want to see that. And you can see that on the packaging. But either way, so a couple examples of GMO organisms are some Atlantic salmon. This is a really good example. Ooh, and I should link a video too. I'll link a video. So obviously Atlantic salmon, we eat salmon, salmon's sold and people buy it, and a lot of people really enjoy it, especially if you're pescatarian and can only eat fish. So obviously, again, going back to just why we genetically modify certain organisms, a lot of the time it's for food resources. So obviously if we're looking at salmon, we want a bigger salmon because bigger salmon means more meat, and that means more fish, and that means we can sell more and we can eat more. So what they've done is they have genetically modified these Atlantic salmon to carry a gene that produces more growth hormone, so that by the time they are fully grown, they completely dwarf the wild variant in terms of size. So obviously that's really good for fishermen trying to sell fish, because then you can sell these for a lot more at market, and then obviously the people are getting more meat back. The risk, and I'll get into this a little bit before tomorrow, but we're going to look at this tomorrow too. The risk with this and the concern is that these fish will eventually, if they're not regulated correctly, they could get into the natural ecosystem and they could mess up the native wildlife and become an invasive species, because this fish obviously takes a lot more food to raise because it's a lot bigger, it's a lot more voracious in its appetite because it has to eat more. So these bigger fish will eat a lot more than these smaller fish, and if these were let out in the wild, they could outcompete the natural variant, which would not be good. Another example are crop-resistant or bacteria-resistant crops. Obviously, there's a lot of discourse with the amount of pesticide use and how much pesticide use is acceptable and the ramifications of using pesticides. So obviously, it's best to avoid pesticides when you can for a number of reasons. So what some people have been able to do is they can breed, or they can not breed. Breed implies that you're just breeding them together, but they've genetically modified certain crops to produce a toxin that will kill bacteria but is a natural pesticide, so it will not harm the rest of the environment. That way you don't have to spray them, you don't have to treat them with pesticides, which cuts down on your pesticide use and the amount of pollution you're releasing into the environment. The issues with this one, which I'll go over a little bit, are issues with concerns of antibiotic-resistant bacteria, which I won't go into too much. I don't remember if we talked about that a lot in this class, but if you took my health, you know what I'm talking about. And then also, this only works with certain crops, so you end up having to spray down most of them with pesticides anyway because the rest of the crops still need pesticides. So is it really a net gain, net loss, whatever it may be? But either way. So real quick, because this is already running a little bit long, I want to go through the potential drawbacks of GMOs, which I already started to talk about. First one I mentioned with the salmon, one major issue with GMOs is competition. If, again, these are not regulated properly, they could get out into the wild and they will start to outcompete because we're breeding them to be most of the time more adapted to their environment because we want organisms that are bigger, better, able to survive more because of just our own personal needs. So if they were to get out, they could outcompete the natural variant. And we've actually seen this a couple times, mostly with plants because plants are a lot less easy to regulate because of how they spread and are able to like get out of containment with their seed dispersal. But if they get into a natural setting, they could outcompete the wild variant and they could drive the wild variant to close to extinction if it's successful enough. Second thing, non-target species. So what this means is this going to have an effect on organisms that we did not want to affect? So for example, if we did that bacterial toxin crop, where it's killing a certain bacteria that would kill the plant, is there anything else that that toxin could affect that we were not expecting? Like any important bug species? Is there any bug that is also vulnerable to that toxin that could be affected by these plants? Or were there certain bacteria on the plant that were necessary for the growth of the plant? For example, certain nitrifying bacteria, bacteria that grow near the roots of plants that help give the plant nitrogen. Are they killing those as well? Because if that's the case, the crops aren't going to do very well. How is this going to affect humans is mostly the biggest one, obviously, that people are thinking about. Like, how is this going to affect me if I eat this GMO and it's been changed in this way? Are there any changes that are going to cause changes in my body? And then the last one are resistant pests. Talking about antibiotic resistance and all that stuff. Again, there's a video here. You can look this up if you want. Either way, normally we'd have more time to talk about this, but it would detract from today's thing. So if you're interested, you can look that up. But either way, so that's it for today. Keep this in the back of your mind. We're going to wrap up tomorrow with some articles talking about the pros and cons of GMOs. But for today, that's really it. I won't post any questions today since we're wrapping up, and I'll be nice. So just look at this, look over this, email me if you have any questions, and just be ready to answer some article questions tomorrow. So bye, guys.